Hi, my name is Ursula Billings, and this is my model of a eukaryotic animal cell. The first part of the cell is the plasma membrane, which is on the outside of the cell. It holds the cell together and it protects it from the outside environment. It's in both animal and plant cells, and it consists of a semi-permeable lipid bilayer that regulates the transport of materials both into and outside of the cell. There are proteins embedded in the plasma membrane that are in charge of this regulation, and they're called channel, transport, cell recognition, receptor, enzymatic, or junction proteins. The nucleus is the largest organelle in the cell, and it controls all of the cell's activities. It also carries chromatin, or the genes that are necessary for the survival of the organism. The nucleolus sits inside the nucleus, and it produces ribosomal RNA. And this type of RNA is necessary for protein synthesis inside of the cytoplasm of the cell. The mitochondria are on the outside of the nucleus, and they're usually called the powerhouse of the cell because they produce a majority of the cell's adenosine triphosphate, or ATP, which carries energy. They have their own double membrane. They can come in any shape, and they may or may not be fixed in one location of the cell. Going back to the ribosomes, they are these small structures shown in red here that are usually attached to the endoplasmic reticulum, or they can be free-flowing in the cytoplasm of the cell, and they're in both eukaryotes and prokaryotes. The rough and smooth endoplasmic reticulum, respectively, are the organelles that synthesize polypeptides and synthesize lipids. The surface of the rough ER is covered in ribosomes, while the smooth ER helps transport molecule-carrying vesicles to other parts of the cell. One part of the cell that these smooth ER vesicles are carried to is the Golgi apparatus. The Golgi apparatus modifies, packages, and transports proteins within eukaryotic cells only. And the lysosomes are the vesicles that are produced by the Golgi apparatus. Their primary job is to digest parts of the cell that are unnecessary or are waste, and when the lysosomes don't work, lipids will build up in the cell, causing the lysosomes to lose their function and for the organism that has them to die. The cilia are the tiny protrusions on the outside of the cell that control the cell's navigation and also help other particles move past the cell on the outside. There's two types of cilia, motile cilia and primary cilia. Flagella are very similar to cilia, except they're longer and they move more smoothly. The extra organelle that I added to my animal cell is the centrioles, which act only during the formation of new cells. So during cell division, they divide and move to opposite ends of the cell, and then they produce spindle fibers that arrange and transport chromosomes. And I made the centrioles out of the penne noodles, which have a very similar long, thin structure. So that concludes the structure of my animal cell. Thank you.